So as requested, I will be talking about adverse outcome pathways in general for the purposes of nano safety assessment. And by the way, I'm Sabina Halappanavar. I'm a research scientist at Health Canada, which is a regulatory institution in Canada. And I'm also an adjunct professor at the Department of Biology of University of Ottawa in Ottawa. So let's begin with what is an adverse outcome pathway. So an adverse outcome pathway is a conceptual construct that portrays existing knowledge concerning the linkages between a direct molecular initiating event and an adverse outcome at a biological level of organization relevant to risk assessment. So it's not really a new concept, as you all know. In the past, it was known by the term mode of action. And the only difference is that the MOAs are chemical specific and AOPs are chemical agnostic. So the main components of an AOP include several key events, key biological events, and a key event that is initiated at the molecular level is termed as a MIE, that's in green here. And then you have series of key events that are intermediate, and they occur at different levels of biological organization, such as cellular, organ, tissue levels. And they connect MIE with an eventual adverse outcome. And adverse outcome itself is also KE. It's a last KE in the process of an AOP. And it represents the clinical manifestation of an adverse outcome. So it's basically a disease KE. So KEs are defined as measurable or observable biological or chemical changes that are essential for toxicity. So we know that there are lots of things happening after the exposure to substances, but those that are essential for inducing toxicity are considered as KEs. And then you have these arrows in between the two KEs. These arrows represent the relationship between the two KEs, which describes the quantitativeness of the response. So how much of KE1 is required to induce KE2 is represented by this particular arrow, or what we call as key event relationship. So basically, AOPs enable systematic and simplified representation and organization of the complex disease processes. In general, AOPs are linear, but multiple linear AOPs may be connected in a network, which demonstrates the complex and intricate mechanisms underlying the disease process. So as you can see here, there may be multiple MIEs leading to a single adverse outcome, or a single MIE may be inducing multiple adverse outcomes. And then even the intermediate KEs may be shared between the AOPs. And networks of AOPs may help strategize screening tests. There are many advantages to developing AOPs, as you see here. They anchor the existing data to a disease process at the appropriate biological level of organization, thereby allowing us to understand what's the right measurement that needs to be made to assess the adverse outcome or to predict the adverse outcome. They also allow knowledge sharing and exchange of expertise by enabling integration of data from diverse sources and methods. And this is very important in case of nanotoxicology, where you need expertise from different scientific fields. By way of doing that, they foster cross-discipline collaborations. They do allow, as I already mentioned in my previous slide, the organization of existing knowledge of a disease process. And more than often we have it in our mind, but it allows us to put it together in a very uniform, harmonized, and modular format, and easy to understand and convey. And so one of the I must mention here that one of the best examples of multidiscipline, cross-discipline collaborations and integration of data from diverse sources, etc., is made by a recent project that's called CHOW, which was initiated by JRC to model the pathology of COVID-19 disease using the AOP framework. I won't go into the details of it, but this was a very fun exercise which is still ongoing, and it involves people from around the world and from all kinds of disciplines, from sociology to clinicians. And it's a true collaborative project, a multidisciplinary project. You can have a look at the website for more details. So the process of AOP development itself is fairly onerous. There are many different ways of going about it. You can start with the MIE if you know MIE and walk forward to an AO, or you can start with an AO backward to a MIE. You also can start middle way out with any KEs that you know of. So there can be case study approaches, or it can be using the data from the high content analysis, etc. There are different types of AOPs. You can have a mini AOP, you can have a putative AOP, you can have a qualitative AOP, or a quantitative or a semi-quantitative. So the quantitative AOPs are the ones that have found some uses in formal risk assessment or formal decision making. But the putative or semi-quantitative or qualitative AOPs have also an important role in providing direction to the research investments and prioritization, as well as they do generate weight of evidence in support of decision making. So once you have synthesized an AOP, you submit it to a database called Aop Wiki, which is the knowledge base of AOPs. And this goes through an internal review process just to see adherence to the AOP principles and guidelines. And then it goes out for the external review by the OECD to make sure the biology represented is accurate. And then once all of this is done, then it goes back to the OECD endorsement by the Working Party of National Coordinators for the Test Guidelines Program and the Working Party for Hazard Assessment. Then, of course, you will have publication of AOPs on the OECD site, as well as any manuscripts that you may want to publish it in. And once the AOP is, of course, developed, then it provides the skeleton that can be used further to validate the concept that is presented here by targeting the measurement of certain KEs, and then seeing how well it predicts the disease, the end disease, and then also generate data for generating quantitative support. Here is just a slide showing the contents of an AOP in the Aop Wiki. So it does contain an elaborate amount of details regarding a disease process. So you can see that there are specific pages for the MIE, KE, and the AO, and apart from describing these different KEs, you also have life stages and taxonomic applications and sex applications, etc. And then you also have overall assessment of the AOP, quantitative assessment of the AOP page, and consideration for potential application of this AOP in a regulatory context, etc. So you can find more detailed guidance and training materials about building an AOP if you're interested in submitting an AOP to the Aop Wiki on the Aop Wiki site. So where are we in nanotoxicology in the context of developing AOPs? There is a recognition and acceptance that AOPs are useful. However, how far have we come in the context of developing AOPs? This is a word computing exercise showing the key events most measured in nanotoxicology studies. And this was part of an OECD WPMN AOP project proposal that was completed last year. It involved multiple OECD country participants. And basically in this slide, what I'm showing you here is the different types of key events that are measured in vitro versus in vivo. And larger the size of a word, larger the number of studies that have measured that particular endpoint. And what we observed is that what we measure in vitro and what we measure in vivo have no connection. There is a large disconnect between these measurements, and that was one of the basic outcome of this exercise that we did as part of the OECD WPMN project. But then there's a pattern that emerges, and we took some of these key events and we tried to understand how do they connect with each other and what does it mean in terms of in vivo. So the first and foremost thing for us was go through this large literature database that we had, consisting of more than 10,000 papers, and then basically identify these various key events in vivo and in vitro induced by nanomaterials and anchor them, basically anchor these in vitro measurements or in vivo measurements to an adverse outcome and in vitro measurements to an in vivo response. So here is an example. The 3 in vitro measurements that we saw in the previous slide that were most measured were cytokine, cell death, and raw synthesis, which were measured to represent inflammation, cytotoxicity, and oxidative stress events in these studies. So we know from the published literature that these are related to each other, these events. And in fact, we also know that together they constitute an underlying mechanism of injury at the tissue level. So then we hypothesize that when we see positive induction of these three events in cells, it may lead to injury at the tissue level. So tissue injury, in essence, can be measured at the cellular levels by the cellular assay targeting these 3 events. And of course, the magnitude of this whole response leading to injury or not depends on the material properties and the exposure doses. But each of these events have multiple measurements, meaning oxidative stress may comprise of separate key events that can be measured in isolation. Also, all these 3 events in an acute scenario may imply signaling. So for example, oxidative stress can be broken down into 3 hub KEs, as you can see here, which reflect the different level of damage. And to observe injury at tissue level resulting in detrimental effects, these 3 events may need to occur. So again, in an acute scenario, just increase in raw synthesis may not be sufficient to see injury at the tissue level, maybe at the cellular level. And the purpose of that cellular level injury could be to signal the defense mechanism and to activate the defense mechanisms. However, in the presence of persistent injury or persistence of the material or repeated injury, eventually the endogenous antioxidant capacity depletes, which will create an imbalanced oxidant and antioxidant status, and that would lead to oxidative modification of biomolecules such as lipids, proteins, and DNA, which then can pose detrimental risk to the organism. So so this is a kind of in trying to understand the biology that was represented in the literature, we had come up with this mini AOP for oxidative stress, and similarly for inflammation later on you will see. And so once we knew what we were looking at, which became very clear now what's there in the literature. The next thing is to go back to the database and collect the weight of evidence to support these key event structure, this mini AOP structure, for example, to establish the KER1 and the KER2, etc. So we found that quite a few studies in nanotoxicology look at these individual KEs in isolation sometimes, which may mean nothing. And many studies do look at these three in together in a study, and so we had quite a bit of evidence to suggest that these are relevant KEs, and we do have enough weight of evidence to support the KEs over here. And another thing that we realized is that there are many different ways to measure the same event. So again, this is the next question. Once you have an AOP in place, the next question is which are the best assays that can be used to measure the KE in a way that it is predictive of the in vivo response. That's the most important thing. One is the measurement of the KE, the best way of measuring it, and also how predictive is that result to predict an in vivo response. So in the end, the outcome of the OECD WPM and AOP project was a mini AOP representing the mechanism of nano-induced tissue injury, and identification of cellular level measurements that would allow its prediction in vivo. So you can go into the details of this study and see how we went about doing this in this particular study. In another recently published study, we established another mini AOP, just like this, using the available data from an experimental setup from a single study actually, describing metal oxide induced in vitro cytotoxicity. So these are a couple of examples showing how AOP framework can be used or implemented to understand the data that you have and identify the KEs that may be useful for an AOP, and then build the AOP. So the next example I have here is the most recent attempt to put together a putative AOP describing the genotoxic mechanism of nanomaterials. Here, a lot of what is known for chemicals was used and repurposed for nanomaterials. So the purpose of this particular slide is to say that we need not reinvent the wheels for nanomaterials. We can use what's there already for the AOPs that are already exist for substance-induced injuries or toxicity, and we can repurpose them. And there is a similar example for lung fibrosis induced by nanomaterials. This is an AOP that I have put together, and this is a putative AOP which has used the existing data for substance-induced lung fibrosis as well as data from the nanotoxicology literature. So it heavily relies on what's known for nanomaterial-induced lung fibrosis as well. This has actually undergone external review by the OECD, and the revisions are being made right now. In addition to that, there are several other individual AOPs representing the inhalation toxicity of nanomaterials. They're all under different levels of development right now. What's interesting is that they share KEs and MIEs, suggesting some common mechanisms underlying the lung response induced by nanomaterials. And these shared KEs can then help form the first-tier testing in a tiered testing strategy. Here in this example, mainly for screening inhalation toxicants. Here is a tiered testing strategy for screening profibrotic nanomaterials that we recommended in this paper here. And basically, when you have an AOP, then different KEs can be targeted, and the early key events in an MIE itself can help with the QSAR modeling, whereas the early KEs can help with the screening assays, and the later KEs, which more or less represent the commitment to disease process or manifestation of the disease itself, can be used as the predictive or confirmatory assays. So here in this, you start off with the pro-inflammatory markers. Depending on the results, you move on to the fibroblast proliferation or collagen accumulation, which is one of the indicator of fibrosis. And then if that also is positive, then you classify a material as profibrotic and prioritize it for further testing with some more advanced testing models. So similarly, this kind of a testing strategies can be built using other AOPs. So AOP building process is kind of iterative in the beginning, in the sense you start off with the toxicological data that is available for you, and you use that and put it in the context of the AOP framework, and then you identify key events and adverse outcomes, etc. Then you put together the whole information in an AOP modular format. Like, so you develop an AOP. Then once you have an AOP, then you design the testing strategies to measure those key events. Then you generate the data using those testing strategies, and use this data again to validate or refine and reorganize maybe the AOP. And so once you have well-tested quantitative AOP, then the data generated with that AOP as a backbone can be used in weight of evidence assessment, informing the regulatory decision making. So in conclusion, I can show a couple of more applications of AOPs. So AOP 173 for lung fibrosis is a putative one. It's not a quantitative AOP, and it's still under construction and revision and refinement. But in its current form, it has already been used to inform structure-activity relationship modeling to predict pulmonary pathology induced by multi-wall carbon nanotubes. So here, the AOP was solely used to help select the parameters for modeling. And here we did not use the MIE because the data was insufficient, but we targeted a key event, an early key event reflecting inflammation for modeling purposes. So this is just to say that you need not have a quantitative, fully developed AOP for it to be useful in your work. And the next one is that although this has not been shown, but this is a possibility. So you can use, well, if there is an AOP that is well constructed, it can help categorize chemicals binning nanomaterials. So the premise of which is that the chemicals that activate the same AOP, trigger the same path to a disease, can be grouped together. And more details about grouping of chemical substances based on the AOP can be found in this particular guidance document highlighted here. And in the end, AOP can also help with the IARA and intelligent testing strategies. So back in 2014, a manuscript written by Stone highlighted the short-term and the long-term needs, or the long-term needs in nanotoxicology, and highlighted various things over here that are circled in red. And looking back at the AOP, we can clearly say that if you have a well-developed AOP, it can really serve as a backbone on which this particular IATA or an intelligent testing strategy can rest. So with that, I would like to say thanks to the organizers again for this great opportunity to present. And the idea here was to just provide you a basic idea of how to go about building an AOP. What are the examples that we have currently in nanotoxicology that we may use? What are the opportunities and challenges there are for developing AOPs? The other aspect of the more important aspect of the AOP is quantitativeness, and that is something that should be covered in another separate study. And there are very good training material available on the AOP Wiki site. Please do visit if you're interested in developing one or learning how to develop one, or how to use an existing AOP, or even to see what are the various different kinds of AOPs under development, even in the field of chemicals. That's the place to go for you. So thank you again.